Hello everyone. So our today's topic is oligopeptides, polypeptides and proteins. So that we are going to discuss today. Okay. And followed by other things. Okay. So what is oligosaccharide? Actually, peptides. So what are peptides? When we have different alpha amino acids, right, attached together by peptide bond. See, this is alpha amino acid. Better to write R1. Wait, madam. Okay, sorry. C double bond O, OH, CHR1, then NHH. Okay, and when it is attached to another molecule, means other alpha amino acid, okay, so H of NH2 and OH of COH, that is removed. So this bond formed means NHCO bond between two alpha amino acids, that is known as peptide bond. This is called peptide bond. Okay, this is called peptide bond. And as two alpha amino acids are attached, that is called dipeptide. 3, then that is called tripeptide. 4, that is called tetrapeptide. On the same line, if we have peptides having relatively shorter chains. Okay, shorter chain means having 2 to 10 amino acid residues. Alpha amino acid residues. So that are called as oligosaccharides. Okay, so these are, what are they? Oligosaccharides. Okay, on the same line like in carbohydrates we have done oligosaccharides, polysaccharides. Like in proteins there are oligopeptides, polypeptides. Now what are polypeptides? Means any peptides having, yes, longer chains. Means these are the peptides having longer chains. Longer chain means, yes, longer chain means, yes, greater than 10 alpha amino acids. So that are called polypeptides. Okay, now out of these polypeptides, those polypeptides having more than, means large number of units alpha amino acids. Okay, any means peptide having more than 10 residues, more than 10 alpha amino acids, that is called polypeptide. Okay, now out of those peptides having yes more than 10,000 unit molecular mass. Okay, or we can say more than 100 units of alpha amino acids, except few. Okay, like insulin. Insulin has only 51 units. Insulin is an exception to the rule. Okay, it has only 51 alpha amino acids. Okay, so means in most of the cases it has more than 100 units of alpha amino acids. So those are called proteins. These are complex nitrogenous organic compounds. Okay, so distinction between polypeptide and protein is not very sharp. Okay, but for the sake of convenience, we should write it as the peptides having more than 10,000 molecular mass. Okay, that is called a protein. Or any polypeptide having well-defined conformational characteristics, that is called protein. Like I gave you example of insulin. Insulin has only 51 alpha amino acid units. Okay, got it? And they are represented as, like if we have glycine, alanine, leucine, isoleucine. Okay, so we can write it like this sequence of alpha amino acids, right? Now these polypeptides are also amphoteric in nature because they have one end carboxyl chain, okay, on and in between whatever peptide linkages, and on the other hand there is NH2 group. So this is also just like alpha amino acid. Protein polypeptides are also amphoteric in character. Okay, because of the presence of terminal those ammonium and carboxylate anion. Because after the shifting of that COH H positive from the carboxyl group, it formed like this. One side there is a carboxylate ion, on the other hand, on the other side it is ammonium ion. Okay, so they neutralize both acids as well as bases, just like alpha amino acids, and they also possess isoelectric point. We have done this. What is isoelectric point? And this at isoelectric point, polypeptides have least solubility. Okay, so we can separate them. Okay, that's it. So this is about oligosaccharide, oligopeptides, polypeptides, and proteins. Now let us classify them. Classification of proteins. Classification of proteins. Okay, now to classify the proteins, we have different ways. The two most widely used classifications, systems of classification are, yes, I think everybody knows. First basis is on the basis of molecular structure. On the basis of molecular structure, proteins have been classified as fibrous, and keep in mind fibrous, see, as the name suggests, fibers. Fiber, you know what is fiber? Fibrous proteins. And second is globular proteins. Okay, now see, fibrous and globular. Okay, so as the name suggests, means fibrous means it is fiber like. Very good. Fibrous proteins. So these proteins, they consist of, yes, they consist of thread like molecules. Okay, and because they are consisting of thread like molecules, these thread like molecules are lying side by side. Okay, as they are lying side by side, and they are held together by hydrogen bonding at many points. Okay, like CO of one and NH of other. So they form hydrogen bond. CO of one, NH of other. They just form hydrogen bond. Okay. So in this way, hydrogen bonding, they are stabilized by hydrogen bonding or disulfide linkage. See, they are stabilized by hydrogen bonds and disulfide bonds. So both are strong bonds. So that means intermolecular forces of attractions are very strong. Okay, and that's why fibrous proteins are insoluble in water. See, intermolecular forces are very strong and they are insoluble in water, and they're also stable to moderate temperature, because if temperature is very high, there is a disruption. Okay, but they are stable. Okay, they are stable on change in temperature and pH, moderately stable. Okay, because if we write very stable, then that may be taken as a wrong statement, but they are moderately stable. Okay, so this is about fibrous protein. Okay, so can you guess one example? Best example is keratin and, yes, myosin. Keratin in hair, skin, even nails. Okay, and myosin in muscles. Okay, any other example? Fibroin in fiber. Fiber means like silk. Okay, so these are the examples of fibrous protein. So fibrous proteins are, first of all, they are stabilized by hydrogen bonding and disulfide linkage. Intermolecular forces are strong. Okay, and they are insoluble in water. They are stable, means moderately stable on changing of temperature and pH. Right? Okay, now come to second point, that is, yes, globular proteins. So see, as the name suggests, they form globules. Globule means sphere type structure. Okay, like polypeptide chains, they form globules. Means polypeptide chains in these molecules folded. Okay, and they are folded in such a way that entire protein molecule is almost looks like spheroid. Its shape becomes spherical. Okay, means they are, first of all, polypeptide chain in these molecules are is folded around itself. Say in globular proteins like thread. Okay, straight chain. Now in this case, globular proteins, they are folded. They are folded. Okay, so when it is folded like this, okay, so because of this folded structure, entire protein molecule is an almost spherical shape. Okay, and folding takes place in such a manner that its non-polar part, means that is called lipophilic, fat soluble part जो है, that is pushed inwards, and hydrophilic part means water soluble, means water attracting. Okay, that part pushed towards outwards, means lipophilic part means hydrocarbon part, that is inward, and this means polar part, that is water attracting, that is outward. This is outward, and lipophilic part is inward. Lipophilic part means water repelling, fat soluble. Okay, and this is water soluble, that you know, hydrophilic, you know. Okay, so in this way, the water molecules, they interact strongly with the polar groups. Hence, globular proteins are soluble in water. They are, because you know, the hydrocarbon part means lipophilic part, hydrophobic, we can say, lipophilic part, that is inward, and hydrophilic part, that is outward, so that can interact strongly with water. Okay, that's why it is soluble in water. Fibrous protein is insoluble in water. Okay, got it? Yes beta. Any doubt up to this? So see, in this case, stability is somewhat less. Okay, and that's why they are very sensitive to change in temperature and pH. Very sensitive to change in temperature and pH as compared to fibrous proteins. That means fibrous proteins are more stable than globular proteins, we can say. Okay, and this class of proteins includes all enzymes. All the enzymes are globular, keep in mind, all enzymes, and most of the hormones. Like best example is insulin. Okay, so even thyroglobulin, that is in, as the names just say, if you write thyro, that means it is in the thyroid gland. Okay, hemoglobin, that is also globular protein. Hemoglobin is used for, yes, for what purpose it is used? You know, transport of oxygen from the blood to muscles. Okay, yes, even albumin in eggs. They are all globular proteins. Okay, and say as they form globules. Okay, so each globular protein has its own characteristic geometry because of the interaction between different sites in the same polypeptide chain. And these interactions are of four types. What I said, they have their own characteristic geometry. And how this geometry is generated? Through the interaction between different sites in the same polypeptide chain. One polypeptide chain is folded back, okay, to give this spheroidal type of structure, okay. And these interactions are: first, disulfide bridging; second, intramolecular hydrogen bonding; say in fibrous protein there is intermolecular hydrogen bonding, but in case of globular proteins, because chain is same and hydrogen bonding is formed, so that is intramolecular hydrogen bonding is formed, okay. Then van der Waals attraction, right? And fourth one is dipolar interactions. Dipole-dipole interaction, okay. So these four types of interactions are there, okay. So globular protein, that may consist of a single polypeptide or many. It may consist of several polypeptide chains, okay. That is called subunits or protomers. What we call them? Subunits or protomers, okay. And each subunit has its own three-dimensional geometry. As I said earlier, each subunit means each polypeptide chain has its own three-dimensional geometry, okay. And when these all subunits are held together by hydrogen bonding, van der Waals attraction, then that gives overall spheroidal shape, okay. So this is all about fibrous and globular proteins. Fibrous proteins are thread-like, globular proteins are spheroidal in shape, okay. Second thing, uh, um, globular protein is fibrous proteins insoluble in water, but globular proteins are soluble in water. Why is it so? Because in globular protein, fibrous proteins, polar groups are form hydrogen bond with each other, but in globular proteins, polar groups are outward, and those hydrophobic, or we can say lipophilic, they are inward, okay. That's why they can interact with water. Then fibrous proteins are more stable because of intermolecular hydrogen bonding. Strong hydrogen bonding is formed, but in case of globular proteins, they are more sensitive to change in temperature and pH, okay. That's it. Now, on the basis of their chemical composition, or we can say nature of products they give upon hydrolysis. First classification we have done, that is on the basis of their molecular structure. Now we are going to discuss, yes, we are going to discuss on the basis of chemical composition, or we can say on the basis of nature of products they give upon hydrolysis. So first is Yes, first is simple proteins. Okay, simple protein means these are the proteins which on hydrolysis give only alpha amino acids, which give only alpha amino acid. Okay, for example, albumin. इसमें होता है albumin in the, yes, white of eggs, or yes, any other, keratin. Keratin in, अभी किया हमने, hair, nails, yes. Now second is conjugated proteins. Simple protein means which have only alpha amino acids, which give only alpha amino acids on hydrolysis. Now conjugated protein means these are the proteins which give even non-protein portion, which give non-protein portion in addition to the alpha amino acids. Now this non-protein portion is called prosthetic group. Okay, what we call it as prosthetic group. Okay, and what is the main function of this prosthetic group? It is to control biological function of the protein. It is to control biological function of protein. Whatever protein that has that prosthetic group, that will control the biological function of that particular protein. Okay, for example, nucleic acids. So nucleoprotein contains prosthetic group as nucleic acid. Okay, lipoproteins, they contain lipids. Phosphoproteins, they contain phosphoric acid residue. Okay, so depending upon the prosthetic group, different names are given to the proteins. Okay, like I said, nucleoprotein. Yes, tell me, in nucleoprotein, what is the prosthetic group? Just write. I have told you just now. Lipoproteins. Yes. Phosphoproteins. Tell me. Even mucoproteins. In nucleoprotein, there is prosthetic group as nucleic acid. Okay. In lipoproteins, yes, it is lipids. In phosphoproteins, there is phosphoric acid. And in mucoproteins, yes, in mucoprotein there is sugars. That is also called glycoproteins. Right? Even chromoproteins like hemoglobin and myoglobin. Okay, there is some coloring matter. Okay. like protoporphyrin, heme group is there, right? Okay. The third category, first is simple proteins and conjugated proteins, and then derived proteins. Under the same heading. So derived protein means these are actually the degradation products which are obtained by the partial hydrolysis of simple or conjugated proteins with acids, alkalis or enzymes. So derived protein means these are degradation products. किसके degradation products? They are obtained by the partial hydrolysis of partial hydrolysis of conjugated or simple proteins. Okay, means they are with acids or alkalis or enzymes. Okay, so in this case what happened, we get protein, from protein we get proteases, then peptones and then polypeptides. Okay, how they derive? First we have protein. Okay, so simple or conjugated, that protein is converted into proteases, then peptones and then polypeptides. Okay, very good beta, very good. Now come to the structures of proteins. So these are the two classifications. First is fibrous and globular, and second is simple proteins, conjugated proteins and derived proteins. Now structure of proteins. So because proteins have a very complex structure, and proteins are biopolymers. They are obtained by condensation of large number of alpha amino acids and which are joined together by peptide bonds. Okay, and it has 3D structure. At the same time, structure of proteins are quite complex. So in order to understand the structure, they are classified into primary, secondary, tertiary, and quaternary structures. Okay, that is the classification. So each level being more complete or means more complex than the previous one. For example, in primary structure, what we do? We try to figure out the sequence of alpha amino acids, because each protein has a specific sequence of alpha amino acids, okay, which are linked together by peptide bonds. So sequence of alpha amino acid of each protein, that is shown by, that is studied under the heading of primary structure, right? Because each protein has a large number of alpha amino acids which are linked together in a specific sequence. So study of that sequence, that is called primary structure, and sequence of amino acids create a different protein. So primary structure is simply, it refers to covalent structure, which include even disulfide bridges of each polypeptide chain. Got it? Okay, beta. And if there is any change in the sequence of a particular alpha protein, then that is called sickle. Like hemoglobin, normal hemoglobin, if it changes into some other defective hemoglobin, okay, so that is called sickle cell hemoglobin. Okay, what it means, that if sequence changes, whole property of the molecule, whole property of the protein changes. Okay, so that means biological property, okay, primarily is due to different structure in their primary structure, means chemical and biological properties. So sequence of alpha amino acids decides this chemical and biological properties of various proteins. That's why sequence of alpha amino acid is very, very important. Okay? If primary structure changes, means sequence changes, whole property of the system, whole property, biological activity changes. Okay? So if there is a destruction in change in sequence, then the biological properties of activity of the particular protein is completely destroyed. Done? So this is the first part. Now second. So keep in mind, primary structure tells us about the sequence, and that sequence will decide the biological properties. Okay. Now secondary structure. So primary structure tells us about the sequence of each polypeptide chain. Okay. Now secondary structure. So in secondary structure, we decide the conformation, or we can say shape of the polypeptide chain. So conformation, which the polypeptide chain assume as a result of hydrogen bonding, that is called the secondary structure. So that means secondary structure tells us about the conformation. Okay, and depending upon the size of the R groups, we have two types of structure, secondary structure, that is alpha helix and beta plated. Alpha helix and beta plated structure. These are the two secondary structures of proteins. Alpha helix, it is like this, means CO of one, NH of the other, that will form hydrogen bonding. CO, NH will form hydrogen bonding. NH, CO will form hydrogen bonding. It is like this. Okay, so in this way, alpha helix is stabilized. And in case of beta plated, we have CO, NH, CO, NH, and so on. on the same line NH, then it is CO, it comes out to be, it is CO, NH, CO, like this. Okay, so in this is called beta plated, means there is intermolecular hydrogen bonding, and in this case there is intramolecular hydrogen bonding. Okay, and they are stabilized by hydrogen bonding, disulfide linkage. Okay, so in this case it decides only conformation of each polypeptide chain, conformation of each polypeptide chain. Okay, now come to the tertiary structure. In tertiary structure, see, tertiary structure of protein represents overall folding of the polypeptide chains. It decides the overall folding. See, in primary and secondary structures, we are dealing with only single polypeptide chain. First is determines the represent the sequence of alpha amino acids in each protein, and second that decide the conformation of each polypeptide chain. Now tertiary structure that represent overall folding of the polypeptide chains. Okay, what it means, that is further folding of the secondary structures. Okay, that means tertiary structure, it refers to manner in which entire protein molecule folds up in 3D space to produce a specific shape. That means it tells us about 3D shape of the molecule, shape of the protein, means all the polypeptide chains, how are they arranged, that is shown by tertiary structure. Okay, at normal pH and temperature, each protein will take up a shape that is energetically most stable. At a particular pH and temperature, each protein has a particular shape, means we can call it as most stable, means energetically most stable shape arrangement. Okay, 3D arrangement. Okay, and this shape is specific to a given amino acid sequence. That means it is called the native shape of the protein. Tertiary structure is actually the native shape of the protein. Okay, so primary structure of protein जो है that dictates its tertiary structure, because different sequence, different shape. So primary structure, that determines the sequence of alpha amino acids. That's why primary structure of a protein that decides the tertiary structure of protein. It dictates its tertiary structure. Okay, so tertiary structure is linked with the primary structure. Okay, and in tertiary structure we have two major shapes, that is fibrous and globular, that we have done just now. Fibrous and yes, fibrous and globular, and these fibrous and globular are stabilized by hydrogen bonding. Yes, then disulfide linkage, then van der Waals attraction, dipole interaction. Means tertiary structure of protein that is stabilized by hydrogen bonding, disulfide linkage, then yes, van der Waals interaction, then yes, dipolar interaction. Okay, so these are the forces which are responsible for the stability of a tertiary structure of the protein. Then last one, last structure, that is yes, dipolar interaction means electrostatic attractions. Okay, quaternary structure. So although many proteins exist as a single polypeptide chain, but there are certain proteins which exist as assemblies of two or more polypeptide chains. इनको क्या बोलते हैं हम? Sub units or protomers. So these sub units may be identical or different, and they are held together by hydrogen bonding, yes, electrostatic interaction. Ashish wrote it. Okay, even van der Waals attraction. So quaternary structure, it determines the number of subunits and their spatial arrangement, means their arrangement in space. So what it tells? Yes, it refers to the determination of the number of subunits. What do you mean by subunits? Number of polypeptide chains in the given protein. Okay, and their spatial नहीं, spatial, sorry, spatial. Okay, means arrangement in space. Arrangement with respect to each other in an aggregate. Means we can say it tells us about the stereochemistry. So say first one, primary structure, tells us about sequence of alpha amino acids. Secondary structure, shape of each polypeptide chain, conformation of each polypeptide chain, alpha helix and beta plated. Tertiary structures, that tells us about overall folding of the polypeptide chains in 3D, means 3D shape of the arrangement of the polypeptide chains. They are of two types, globular and fibrous, and they are stabilized by hydrogen bonding, van der Waals attraction, electrostatic attraction, that dipolar interaction means disulfide linkage. Okay, and quaternary structure, that tells us about the stereochemistry of all the subunits, all the protomers or all the polypeptide chains. Okay, and it refers to the determination of the number, see, number of subunits and their arrangement in space, right? So this is all about shape of the proteins. Okay, so let's stop here. We will continue our lecture tomorrow, right? So bye bye. Any doubt up to this? Yes, tell me. If you have any doubt, then you may ask. Yes, beta. Any doubt? Okay, bye bye, beta.